Well, now we will have the presentation of this famous analysis that I've been earned for, and everyone has been writing about this research and uh, claiming this research even before this was made known and certified. There was a press conference yesterday at the Chamber of Deputies. No newspaper talked about it. I don't think they're going to discuss about this even tomorrow. Anyway, the fact that we are in streaming, that there are televisions from Belgium as well to follow our works with such a qualified audience, and I would like to tell you some figures as well. We have 50 guests, and we also have 260 biologists, physicians, pharmacists, researchers, university professors that I would like to thank, who belong to this science that is not honored and credited, but nevertheless represent the backbone of research in Italy. Thank you. So I start by making an important preamble. I've been a consultant of the Parliamentary Investigation Committee on Depleted Uranium. I was a consultant for this committee. Among the members, there was also the present Minister of Health, Grillo. And as a consultant on military vaccination, I was involved in a great work of review of the literature, but also studying of registration dossiers for the confidentiality of vaccines for military use. And I have to say that military use vaccines are the same that are administered for civils. So what has been shown, that has been written also in the final report, is that in the end there are some doubts, some things that still need a further investigation, especially as regards quality. It has been said that the committee has as its target, its primary goal, among the many objectives that it had set, also the one to analyze the composition of vaccines. So the study of quality of vaccines through an analytical investigation was already scheduled in the committee. So what I'm introducing today is simply progress of the work of the committee that it was not possible to start and of course not even to finalize due to reasons that went beyond the commitment that the Parliamentary Investigation Committee had taken. So I have to say that for the beginning that it is a quality study. We all know that unfortunately there is a slogan that is reiterated on the media, that vaccines are clean, safe and effective. What we are trying to investigate regards, are vaccines clean? Question mark. So it's related to quality. So the first study hinges on the goal of this investigation. Today we are witnessing this step, assessment of the compliance of composition of vaccines as indicated in the datasheet of vaccines, so going back to verify whether all the components are there, and then the screening of first level. The first level screening of contaminations, chemical, proteic and gene material contaminations. In the registration dossiers, we had highlighted that in the end, actually, there were some chemical contaminations and protein contaminations. Instead, the indication on the study of the gene material was absolutely missing, for example. So now we have just finalized the first level screening, and so we are in a phase in which data are still probable, and we have the first confirmation data. So what you're going to see is data that have been obtained through the analysis performed in the laboratories that have validated methods, certified methods, also for the equipment that they use. These laboratories are working for third parties and so release analytics certificates. And so we have performed in-depth studies as soon as we got the results to try and understand in a better way the meaning of these outcomes that were obtained. in a short time, in a short run, I hope from next month, so we're going to perform the second level study. So we're going to characterize the compounds that have the highest problems for safety and effectiveness, and then we're going to make the final interlaboratory confirmation test. So we have decided to disseminate data from the very beginning, also for reasons of transparency. If we waited to have the peer-reviewed publications, it would be necessary to take a longer time. Instead, we decided to make this data available, even though there were many polemics regarding this, to allow everyone to know anytime the procedures and the progress of our investigations. So this is, roughly the goal of what we are doing. The technologies that we have used by now are of two kinds. We have a technology that is used for the analysis of the genetic material, and we used a next generation technology that is called next generation sequencing. This technology is currently the most sensitive and specific to analyze the content and the DNA and RNA antigen content, and also in any contaminants. So the second technology instead is the mass spectrometry that is highly sensitive and highly specific. And I have to say that this method was selected and acquired by the FDA precisely for the study of contaminations, and the protocols are applied that are used for the forensic analysis. So the procedure is extremely precise. So by now we have succeeded in completely analyzing, so from the profile of the contaminations for vaccines. Infrarix Hexa, it's an hexavalent of Glaxo. Priorix Tetra, that is still belonging to Glaxo. Infrarix Hexa has been registered at the European level, so the AMA, the European Agency for the registration of drugs, is responsible for that. Instead, Priorix Tetra is a drug that has been registered in Germany and was acquired as a registration also in Italy for the mutual recognition. The Hexion is another hexavalent, and Gardasil 9 is the vaccine for papillomavirus. These are both registered at the European level. I'm stressing this because there is, a rebalancing of responsibilities as regards these vaccines between the IFA and the EMA. We have to say that actually when the state acquires a product to sell this product on its national territory, it is always, the regulator of that country who is in charge of being, liable for the safety and the effectiveness of the product and the quality of the product that is publicly available. Quite briefly, this is the composition of Infrarix Hexa. As you can see, underlying that, here we have a very high number of antigens. Some of these are detoxified toxins with formaldehyde and glutaraldehyde. Then we have the polioviruses that we're going to study later on. There are no viruses in the vaccine, but there are just proteins, the famous antigen D protein. We have a recombinant vaccine of hepatitis B and a conjugated vaccine of the Haemophilus that is linked to a carrier protein, and there is which is a tetanus, and there is aluminum hydroxide, and the cells that are used with this tetanus toxoid to propagate the polio are immortalized cells. These are Vero cells lineage, which are carcinogenic. So I apologize. I just wanted to show you that in the data sheet, there are anyway the indications of some residuals like informaldehyde, neomycin, and polymyxin. The producer has to signal this because they can bring to hypersensitivity and safety problems. In the data sheet, it is not written that there is a preservative. It is just in the discussion of the EMA. So anyway, it is reported as a product. So the first analysis that we carried out was performed precisely on the genetic part. So we wanted to ascertain whether our vaccine contained the poliovirus. So the first result was exactly what we have found. So we have asked to the EMA why we couldn't find these viruses of polio, and the EMA promptly replied saying that the treatment with the formaldehyde degrades all the genetic material, including also viruses of polio. And so this treatment has to be performed because, unfortunately, in the vaccine, there are also some genetic materials of the viral lineage cells. So there is a guideline which states absolutely that in vaccines there must not be DNA coming from this cell lineage, which is very dangerous. And of course, we didn't find it actually, but we found some traces coming from the viral cells in quantities that are below the detectability threshold of the tool, which is very sensitive. So we can say that it is not a contaminant of the vaccine because it is highly degraded, and so it does not create problems for safety. Instead, what raised many concerns were the proteic analysis, the chemical analysis as well. So we decided to keep a minimum threshold that was relatively high for the tool, so that we could state that what we have found is non-residual. So what we find here, we can state that these are actually the components of the vaccine. We cannot talk about residuals because residuals in the end actually should be below 10 nanograms, at least according to the guidelines. So what we found is 65 signals. So the signal identifies a compound when the structure, the molecular weight is very low, very limited. So when we talk about big molecules, a big molecule can even have 3 signals. So this is overestimating a bit the number of compounds. So let's say that roughly here we would have about 2/3 are actual compounds. So only 35% were identified. So we have 35% of compounds that are candidates because they have been identified through a data bank, and the other 65% that we do not know absolutely what it is. We found also 16 peptides, so amino acid sequences that are very small coming from the bacteria world, so these are potentially allergenic. And we found in these toxins, in these chemical contaminations, 8 molecules as well that are classified as toxins. Toxins, of course, have to be below the detectability limit because these are extremely dangerous, so they should not absolutely be contained in the vaccine. So the factor that raised many problems was the lack of the identification of the antigen.
The analysis certificate, and the researcher told me that there were no proteins inside the vaccine. But there were 10 protein antigens, and they said no, we haven't found them. So we had to re-examine the vaccine, and we discovered that instead of the antigens, there was a macromolecules which has protein functional groups, and so it acts also as a specific reactive for proteins, but it doesn't have the features of a protein. So it has a chemical composition and chemical and physical composition which cannot be protein. And so we have included everything. So the fact that it was not cut was a feature of this molecule, and it was a problem of method. So we have decided to say that antigens were not detected. So this causes a problem, an efficacy problem, because for a protein to generate effective antibodies, it must have the possibility to interact. To be split within immune cells in order to be exposed to the surface and create antibodies. If it is not possible to digest this substance, it will not be digested inside the cells, and so we wonder how this can generate protective antibodies. So this is not a statement, this is a doubt, this is a question we raise to producers and regulatory agencies, because we want to understand how this macromolecule can be referred to as an antigen capable of forming protective antibodies. The second vaccine we analyzed is Prerix tetra. We have four viruses: measles, rubella, chickenpox, and mumps. And we have verified all of them. This is the cell line. The cell line comes from a male fetus of 14 weeks, which has been cultured in 1966. So starting from 1966, that cell line is kept in culture for the production of viruses. So it is quite an old cell line. Dr. Deischer will explain in her presentation which are the problems which the presence of a potential DNA in the cell can entail. Because in 3 different lots, we have seen a big variability in the content of this contamination, which was a waste of the manufacturing. But because of the quantity, you can see 3.7 micrograms per dose, it is a real component of the vaccine. So this is almost the quantity of DNA present in 370,000 fetal cells, and so a huge quantity which caused a lot of problems in the study of antigens. We had to work a lot in order to find our viruses within this highly contaminated material. So you can see that based on the DNA analysis, we have seen a total DNA, 80% is human. We also found chicken DNA, proteobacteria in variable quantity. In the RNA, so the other type of genetic material, we also found almost 80%. We found other materials coming from other substances, and so all the different tests have been performed in order to avoid any lab contamination. Of course, we will have a further confirmation when we will perform the intra-lab analysis. As for the viruses, chickenpox, mumps, measles, and rubella, so they were sequenced, identified, and unfortunately we had a big problem with rubella because we thought it was a method problem. We could not find it, so we had to do a big sequencing. We reached 260 million sequences analyzed, and we found 114 sequences. So rubella is there, but the quantity is about just one viral contaminant. While we were searching for rubella, we also found contaminating viruses, and so a retrovirus K, the leukemia virus, equine leukemia virus, avian leukosis virus, and virus. So these are retroviruses, and the danger feature of these viruses is well known, because if we think we can study the cancerogenic effect of these viruses, we need long-term studies, which I haven't seen so far. So there is a big problem concerning safety in these viruses, which has to be investigated. If we do not know what they do, EMA says they must not be there. So the EMA doesn't say, well, that's okay. No, the fact that they do not exist is not okay for the EMA. Then chemical analysis. Here you can see a list. We have found a higher number of signals with an average of 30% known. So 70% we don't know what to do to understand what 70%, the remaining 70% means. We have found a set of substances, quite peculiar substances. Have listed only a few of them because the list is very long, but there is probable herbicide, antibiotics, and crossed contamination. So probably in the vaccines there are contaminations from drugs which are produced in the same manufacturing plant, and so traces of the other drugs end up in the vaccines. So these are not traces because they are known residual quantities. So it means that there must be a check on crossed contaminations which will have to be performed. It will be good to have a response on this. The other hexavalent vaccine, Axion, was the object of a long debate. We didn't find antigens in the first vaccine, and so some scientists said that the method was wrong. We applied the same method, so we did the same things on this vaccine, Axion, and we found antigens. We found 3 out of 6 antigens, so it means that 6 of them were sequenced and the other 3 were not identified. So they probably ended up in the 70% of non-identifiable compounds. So the method is consistent, and so it's relevant. In Axion, we found a higher number of contaminations, chemical contaminations. The number of chemical toxins is higher, and they must not be there. The metagenomic analysis is quite important. Unfortunately, this vaccine has DNA contamination, so it means that the formaldehyde treatment is not so strong, and so there is the possibility for contaminations which are dangerous, and actually we have to investigate this dangerous feature. So we have a contamination from bacteria which are used for the production of toxins. So this DNA is immunogenic, and so there can be allergies and immunity problems. This material is associated with aluminum. So aluminum has the feature to make RNA, DNA, and proteins very resistant to degradation. So I would expect these elements to stay in the organism for a long period of time and to stimulate a toxicity which unfortunately has not been investigated appropriately. DNA of monkeys coming from viral cell lines, which should not be there, but we are talking about very neglectable quantities, but even if we have 10 nanograms of DNA, we already have 1,000 carcinogenic cells, and so 10 nanograms is the limit which has been set by the EMA for these vaccines. But we're talking about cancerogenic cells, and so it is quite a danger. We found other viruses, the phage of tetanus, carrier used for cloning. We found RNA coming from cultures used for the production of toxins, and we also found RNA of monkeys, and this shows that the treatment with formaldehyde is not so strong. Then we have the last vaccine, Gardasil 9, which is a totally engineered vaccine, so it is produced through genetic recombination. It has adjuvant, and as you can see, there are not many problems. The results of the metagenomic analysis, well, in my opinion, and these are data which has already been manually confirmed, so second level data, because it has been confirmed. So we have bacteria quite in a big amount. The quantity of total DNA is not so high. We are at the limit with traces, so these are not components of vaccines, these are just residual elements. So bacteria, human DNA, and mice DNA, so contaminants coming from other cellular lines, because otherwise I would not understand why we have the mice DNA. Then we have found the virus of papillomavirus. Inside the papillomavirus virus, a fragment of DNA, which is a contaminant, and there are already some literature studies on these fragments. These fragments are associated to aluminum, and in the research carried out by Professor Lee, we have seen that these fragments stay in the organism for a very long time, and they can cause allergic reactions. They can stimulate cytokines in the cells, and so very important inflammatory processes. We also have phages and viruses which should not be there, retroviruses also in this case, and potentially they can integrate with the DNA. And in the RNA analysis we have synthetic material from artificial sequences which are produced by genetic recombination. So these synthetic materials, can this synthetic material do something and be dangerous? Maybe they are dangerous because they are not degraded, so they stay in the organism, and but we don't have verified consequences. Then we have yeast, which causes allergies. We also found the virus of the equine anemia, which is a carrier from genic therapy. So it might be a potential cross-contamination. So even in this case, we don't have a great control. I read the EMA guidelines, but no mention is made to this contamination, which is not assessed. No reference is made to retroviruses. We have an FDA guidelines, which is quite recent and forces producers to check the presence of these viruses in vaccines. And so the regulatory agencies evolve through the knowledge. And so what this information we provide is a knowledge which has to be disseminated, because we are using very important technologies, state-of-the-art technologies, which have not been validated yet in labs in accredited labs. And so these studies are studies which are carried out in the research and development phase, because the analysis of the lot is done on known compounds which have been agreed upon by the EMA and the producer, because there can be safety problems, etc. But these are known compounds. In this case, we have seen that there is a huge quantity of unknown material. We don't know where this material comes from. We don't know the structure of this material. So, also in this case, the same as for the chemical protein-based analysis, we have identified 7 out of 9 antigens. Maybe it is chemically modified, just as the molecule of the other vaccine from the aluminum, which is in the vaccine. We don't know. The number of signals is very high, 22% known, 10 toxins, and now I'd like to give a few remarks on the importance of the results achieved. If we talk about quality, we do not talk about the safety and efficacy of vaccine, because this requires specific studies, but we can raise questions and doubts and problems. If we don't identify vaccine antigens, have a doubt on the efficacy, which has to be analyzed. If we find chemical contaminations, which are real compounds and components, then there is a safety problem related to the toxicity of the dose, because if we have a substance equal to 1 microgram, having a 1 microgram of 100 substances is different in terms of, has a different impact in terms of toxicity. And in the same way, if I have a high percentage of unknown substances, what shall I do? The EMA states that if we don't know what it is and what it does, it has to be close to 0, and so we have to perform cleaning processes which allow me to eliminate these materials. But the presence of residual elements from the manufacturing, what type of effect does it have on the DNA? If a contamination causes a potentially carcinogenic effect, I see this in the long term, and so there is the need to assess safety in the long term. So as of now, based on these results, which are quite preliminary, so we are assessing the results and confirming these results, we can say that if we cannot assure the validity of these doubts, we cannot ensure the safety and efficacy of vaccines, and so quality is the first point. Excuse me? I'm sorry, we cannot hear if they don't speak on the microphone. So I have concluded. Hope I was clear enough. Thank you.